Okay, so the focus of today's lecture is the brain and the cranial nerves before we move on to the spinal cord and the spinal nerves next. This lecture will have quite a strong focus on anatomy, so looking at various structures and regions of the brain, but also how these structures work and their major functions. Before we jump into the lecture, I just wanted to point out that given the strong focus on anatomy, most of the lecture slides are going to contain lots of pictures and very few words. In saying that, I have summarised many of the structures that we'll look at today, along with a bit of a brief function, so don't stress about taking a whole heap of notes. I would rather you look at the images as I speak, and then if you need to go back to jog your memory about what each structure does, I've written these notes up for you and I've posted them on Canvas. So learning objective number 1 is going right back to the start and having a look at the embryonic development of the brain. So for this learning objective, I'm going to play you a really short video. This video comes from the YouTube creator 2 Minute Neuroscience. I highly recommend these videos. They explain a number of topics relating to the nervous system really simplistically, but also really scientifically accurately, which obviously is important. I also really like how they will draw along with the process that they're explaining. If you want to watch this video again or check out any of their other videos, I have popped this link on Canvas as well.
Welcome to 2 Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss early neural development. The development of the nervous system begins at around the 3rd week of embryonic development, when an area of the ectoderm, or the outer tissue layer of the embryo, thickens and forms what is known as the neural plate. This plate begins to fold inward, forming a groove called the neural groove. The sides of the neural groove, known as the neural folds, begin to come together. At the end of the 3rd week, the folds will begin to fuse together. By the end of the 4th week, they have completely fused together to form the neural tube, which will eventually become the brain and the spinal cord. As the neural tube closes, bulges and bends begin to appear and they gradually become more noticeable. During the 4th week, there are 3 of these bulges present. They are called the primary vesicles. They are the prosencephalon, which will eventually form the cerebrum, the mesencephalon, which will eventually become the midbrain, and the rhombencephalon, which will eventually become the rest of the brainstem and the cerebellum. The end of the neural tube will form the spinal cord. As the brain continues to develop, 2 of these vesicles further subdivide to form secondary vesicles. The prosencephalon forms the telencephalon and the diencephalon. The telencephalon will become the cerebral hemispheres. The diencephalon will eventually consist of the thalamus, hypothalamus, and other structures. The mesencephalon does not subdivide any further and will become the midbrain. The rhombencephalon will subdivide into the metencephalon and the myelencephalon. The metencephalon will become the pons and the cerebellum, and the myencephalon will become the medulla. The spinal cord will still form from the end of the neural tube. Next, the neural tube will continue to develop to look more like the brain. The telencephalon will grow more rapidly than other parts of the tube, and by 11 weeks, the brain will have a similar shape as to what it will at birth. Although it will continue to develop after birth, the brain at birth is structurally similar to a fully developed brain.
Okay, so that video described the development of the brain in utero really well, but I just want to note that I'm not too fussed about you remembering too many of the specific details, the names of the various folds and the grooves. Rather, I want you to be aware of the general development of the brain and the spinal cord. So it develops from the outer layer of the embryo. This tissue forms a neural tube, and from this neural tube develop all the other regions and spinal cord of the brain. You might have also noticed how many of the different regions of the brain as it's developing end in the suffix encephalon. So cephalic or cephalon refers to head, encephalon means brain, and then the prefix of each region denotes the location or the order of development. So for example, the mesencephalon is the part of the brain that develops in the middle portion of that neural tube. It eventually becomes our midbrain. So mes means middle and encephalon means brain. So it's that middle part of the brain. Now to quickly recap the video, the brain and the spinal cord begin to develop in about the 3rd week of gestation. The outer layer of the embryo, which is called the ectoderm, thickens and becomes our neural plate. So that's this region along here. The neural plate then folds inwards and forms a longitudinal groove. So this is our neural groove. The raised edges of the neural plate, so here and here, are our neural folds. As development continues, those neural folds increase in height and then they eventually come together to form the neural tube. As development continues, the front part or the anterior portion of that tube will develop into 3 enlarged areas, which we call our primary brain vesicles. Again, I'm not too worried about you knowing each of these names. I will not assess you on that, but just for interest's sake, the prosencephalon is our forebrain, the mesencephalon is our midbrain, and the rhombencephalon is our hindbrain. As development continues, we then have our secondary brain vesicles develop. So the prosencephalon develops into 2 more vesicles, our rhombencephalon also develops into 2 more vesicles. Our development continues, and particularly the front or the anterior part of that neural tube expands and grows at a much faster rate than the rest of that tube. This starts to produce that shape of a brain that we recognize in a newborn or in an adult. And then at birth, structurally, our brain is very similar at birth to what it is as an adult. Of course, there's lots of development and strengthening of the neurons and the synapses to occur as we develop and grow, but structurally, it looks quite similar. So learning objective number 2 is to describe the protective structures of the brain, and there are 3 that we're going to focus on today: the cerebral meninges, the cerebrospinal fluid, and the blood-brain barrier. So starting with the cranial meninges, the meninges are 3 layers of membranes that surround the brain and the spinal cord. Although they are continuous with one another, so it's one long piece of connective tissue, the meninges that surround the brain are called our cranial meninges, and those that surround the spinal cord are called the spinal meninges. The 3 layers that make up the meninges, from most superficial or most external to the deepest or most internal, are the dura mater, the arachnoid mater, and then the pia mater. So taking a look at these and the other tissues that surround and protect the brain, we'll start here on the left at the most superficial. So first off, we have our scalp, which is just the skin, our hair, if you have hair. We then have an epicranial, so epi means on top of, aponeurosis. So an aponeurosis is just a thick layer of connective tissue. We then have the periosteum of the skull, so that membrane surrounding the bone of our skull. We have the skull, the bone itself. And then we have our 1, 2, 3 layers of the meninges. So the first layer is our dura mater. In Latin, this translates to tough mother, and this refers to the thick leather-like texture of this layer. We actually have a brain in the wet lab with the dura mater still attached to the brain, so if you'd like to have a look in your lab, please make sure that you ask your tutor. This layer is composed of thick, strong, dense, irregular connective tissue. It surrounds the entire brain and spinal cord, and it actually holds the large veins or collection of veins that carry blood from the brain back to the heart. Moving internally, our next layer is our arachnoid mater. So arachnoid refers to spider. It's referring to the spider web-like texture of the collagen and the elastin fibers in this membrane. In the space between our arachnoid and our pia mater, we have a space called the subarachnoid space, so here. And this is where our cerebrospinal fluid will sit, which we'll talk about next. We also have little extensions of the arachnoid mater that project into that subarachnoid space. These are called the arachnoid villi, and these help reabsorb the cerebrospinal fluid back into the blood so that it can be refreshed. Most internally, or the deepest layer we have, is our pia mater. So our pia mater translates to tender or to delicate mother in Latin. And this is a very thin, transparent layer of connective tissue that it adheres directly onto the surface of the spinal cord and the brain. So it actually dips into all these bumps and grooves. It's very difficult to actually distinguish it from the brain or the spinal tissue itself. Within the pia mater are many blood vessels that supply oxygen and nutrients to the brain. So putting all of that down in words for you, the dura mater is the outermost of the three meninges. Dura mater translates to tough mother in Latin, and it encloses or it supports the collection of veins which drain the blood from the brain. Our arachnoid mater sits between the dura and the pia mater. It's spider-like, it looks a bit like a spider web. It forms the arachnoid villi, which reabsorb the cerebrospinal fluid. And our cerebrospinal fluid circulates in the subarachnoid space, which is a space between the arachnoid mater and our pia mater. So our pia mater is the innermost layer of the meninges. It translates to tender mother. It adheres directly to the surface of the brain, and it's permeable to water, solutes, and blood vessels. So those substances still need to reach the brain tissue. So moving on to our cerebrospinal fluid, our cerebrospinal fluid, which sometimes you will see written as CSF, is a clear liquid composed mainly of water that surrounds the entire brain and the spinal cord, sitting in between that arachnoid mater and the pia mater. The function of the cerebrospinal fluid is to protect the brain from both a physical injury and a chemical injury. So firstly, it acts as a shock absorbing medium, so it protects the brain from mechanical or a physical injury. The fluid sits between the two deeper layers of the meninges and acts a bit like a cushion. So it stops the delicate tissue of the brain and the spinal cord from coming up against the hard bone of the skull or the vertebral column when the body is knocked or jolted. This fluid also allows the brain to float within the skull. So this is important because our brain is actually quite heavy. It weighs approximately 1.5 kilos. And so we don't want that under delicate tissue of the brain resting on the base of the skull. So by having this fluid, the brain can float and that tissue is protected. Secondly, cerebrospinal fluid has a homeostatic function. And this is because it allows certain substances to move from the blood into the cerebrospinal fluid and then into the brain tissue, and vice versa. So we can transport nutrients, waste products, and hormones via our cerebrospinal fluid. It also helps maintain an appropriate osmotic pressure, which is related to hydration. It helps maintain pH, and also our brain temperature. Thirdly, more or less cerebrospinal fluid can be produced or reabsorbed to adjust intracranial pressure, which is just the pressure within the skull. So if the production of cerebrospinal fluid is increased, there's more fluid within the skull that increases intracranial pressure, and that actually inhibits the flow of blood to brain tissue. If we go the other way, so if the production of cerebrospinal fluid is reduced, or say we reabsorb more cerebrospinal fluid than we're producing, there's less fluid in the skull, which means less pressure, and it's easier for that blood to flow. So this is why anytime there's an increase in pressure in the skull due to an injury or a bleed, it can be really dangerous. That increase in intracranial pressure actually inhibits the flow of blood to various regions of brain tissue. So our cerebrospinal fluid is produced by choroid plexuses that sit within the 4 ventricles of the brain. The ventricles are the structures here highlighted in blue, and they're simply interconnected cavities within the brain tissue. We have a lateral ventricle in each hemisphere, so you have one here and then one on the other side. These are referred to as our first and second ventricles. Our third ventricle sits in the midline of the brain here. It's quite a narrow cavity. Our fourth ventricle sits down towards our brainstem, heading down towards our spinal cord. So cerebrospinal fluid is first produced in these lateral ventricles. It then flows down into our third ventricle, where more cerebrospinal fluid is produced. Flows down this little tube here, which is our cerebral aqueduct, into our fourth ventricle. More cerebrospinal fluid is produced here, and then that fluid flows down the central canal. So the central canal is just basically a hole that runs the entire length of the spinal cord. From the central canal, the fluid then exits the spinal cord and moves into that subarachnoid space between the arachnoid mater and the pia mater. It runs all the way around the outskirts of the spinal cord and then all the way around the outskirts of the brain. So our cerebrospinal fluid is produced by a choroid plexus, which sits within our ventricles. That's this kind of funny looking textured shape here and here. A choroid plexus is simply a network of blood capillaries surrounded by ependymal cells. So you'll remember we talked about ependymal cells when we looked at our neuroglia. So taking a closer look, here we have our capillary, here we have our ependymal cells surrounding that capillary. So our ependymal cells are cube-shaped cells that tightly adhere to one another. Because of this really tight junction or tight barrier, only selected substances, which is mainly water, can actually flow from the blood through those ependymal cells into the ventricle to produce that cerebrospinal fluid. Substances can also move back the other way, so from the cerebrospinal fluid through the ependymal cells and back into the blood. The ependymal cells also help with the movement or the circulation of our cerebrospinal fluid. So you'll see that our ependymal cells have cilia, or these little finger-like extensions on the external side of the cell. And as the heart beats, our arteries will vibrate, and this vibration causes these cilia to move. So the movement of these cilia actually helps move the cerebrospinal fluid through each of the ventricles, through the central canal, and then through that subarachnoid space. Before we move on, I also just wanted to point out the arachnoid villi that I mentioned when we talked about the meninges. So they're these little extensions here. Remember, they're extensions of that arachnoid mater. They project into our subarachnoid space and they help reabsorb that cerebrospinal fluid after it's gone through that pathway around the spinal cord and the brain. So the last protective structure that we'll look at is the blood-brain barrier. And the blood-brain barrier is a term used to describe the unique structural and physiological properties of blood vessels in the brain. So the blood-brain barrier consists of endothelial cells that are joined really tightly together. So typically we have a little bit of a gap between our endothelial cells so we can have substances diffuse in or out of capillaries. In our blood-brain barrier, these are nice and tightly joined together. In addition to the tight junctions of our endothelial cells, we also have feet, or the processes of our astrocytes, surround those blood vessels and surround those endothelial cells. And this helps produce kind of like a secondary boundary around that vessel. The blood vessels in the blood-brain barrier allow some substances to pass through. So things like water, glucose, oxygen and carbon dioxide, and also alcohol, which explains why you get so affected by alcohol. But then it also restricts, and probably most importantly, it restricts the passage of others. So it holds back proteins, it will hold back pathogens, and it also holds back many drugs. So while the blood-brain barrier is a good thing, this can create issues when we have illness or disease that affects the brain, because the blood-brain barrier will actually prevent some medications and from some treatments from reaching that affected brain tissue. Physical trauma, certain toxins and inflammation can cause irritation to the blood-brain barrier, which causes that barrier to become more permeable or let things through more easily. And so clearly this increases the risk of a harmful substance entering the circulation of the brain and affecting the brain tissue. So to give you a bit of a visual of what the blood-brain barrier looks like, I have another short video for you. This is also from 2 Minute Neuroscience.
Welcome to 2 Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the blood-brain barrier. The blood-brain barrier is a complex that surrounds most of the blood vessels in the brain. It acts as a barrier between the bloodstream and the extracellular space of the brain, allowing only certain substances like water, oxygen, and small lipid-soluble substances to easily cross from the blood into the brain. This prevents toxins, pathogens, and other potentially dangerous substances from crossing from the circulatory system into the brain. It is thought that the central components of the structure of the blood-brain barrier are the tight junctions of endothelial cells, the cells that make up the interior surface of blood vessels. In other blood vessels throughout the body, there are small spaces between these endothelial cells. Small blood-borne substances can pass through these spaces and into surrounding tissues. The endothelial cells that make up the blood-brain barrier, however, are fused tightly together to form tight junctions that restrict diffusion across the blood vessel lining. Glial cells called astrocytes also have projections called astrocytic end feet that extend to the walls of blood vessels that are part of the blood-brain barrier. Astrocytic end feet often completely surround blood vessels in the brain and are thought to play critical roles in the formation of the blood-brain barrier. For example, they seem to be involved with signaling that prompts endothelial cells to form the tight junctions necessary to create the blood-brain barrier, and they are believed to have multiple functions involving the maintenance and regulation of the blood-brain barrier.
Okay, so moving on, learning objective number 3 is to locate and describe the gross anatomical structures of the brain. And when we say a gross anatomical structure, we mean the big structures, the big regions, the parts of the brain that you can see with the naked eye. So we're going to break the brain down into 4 regions, and each of those regions will have smaller structures that we'll be taking a look at. So first we have the cerebrum. So that's this outer portion all the way along here, the pink folded wrinkly area. And we'll be looking at the cerebral cortex, the corpus callosum, and then the different lobes of the brain. Next is the diencephalon, and that's this central core of tissue here. It contains the thalamus, the hypothalamus, and the epithalamus. We then have the brainstem. So the brainstem is this section down here. It will then continue on into the spinal cord. Here we'll look at the medulla oblongata, the pons, and the midbrain. And then lastly, we have the cerebellum. So that's this little part here that's often referred to as the mini brain. So starting with the cerebrum, which is the entire area here, here. The cerebrum is the largest and the uppermost portion of the brain. It's divided into a left and into a right hemisphere. The cerebrum allows us to complete what we call our higher order functions. So it allows us to do things like reading, writing, thinking, to make calculations, compose music, to remember the past, plan for the future, and imagine things that have never existed. Our higher order functions generally fall into 2 categories, either cognition, so this includes our intellectual function, our memory, speech, language, planning, and decision making, and then our behaviour. And this is typically a manifestation of these cognitive functions, but influenced by things like our personality. So the cerebral cortex is one of the structures within the cerebrum, and it makes up a thin layer of grey matter. So our cell bodies and other unmyelinated parts of an axon. You can see it here stained in dark purple, the very outer layer of the cerebrum. Although it's only 2 to 4 millimeters thick, the cerebral cortex contains billions of neurons arranged in layers. During embryonic development, when brain size increases rapidly, the grey matter of the cortex grows much faster than the white matter underneath. So this lighter purple colour here is all our white matter, which is our myelinated axons. Because this cerebral cortex grows so much more quickly than the rest of the brain, it's what gives it this kind of folded and wrinkly appearance. Now, when we're looking at particular structures of the brain, there's particular things that we look for to locate these structures. The folds or the bumps in the brain are what we call gyri or gyrus as a singular. The deep grooves, so for example this one here or this one here that separates the left and the right hemisphere, are called fissures, so F-I-S-S-U-R-E-S. And then our shallower grooves, so for example here and here, are what we call sulci or sulcus as a singular. Now, as I said, the most prominent fissure in the cerebrum is the longitudinal fissure, so that's the one that separates the left and the right hemisphere. The cerebral hemispheres are then connected internally by the corpus callosum. So the corpus callosum is a broad band of over 200 million myelinated axons. The corpus callosum is what allows communication from one side of the brain to the other side of the brain. If the corpus callosum is severed, although you will still be able to survive, the brain's hemispheres will not be able to communicate properly with one another, and you can have a loss of a range of functions. So for example, changes to your visual perception, where you can see slightly different images in each eye, changes in your ability to produce and understand speech, as well as changes in memory. Having said that though, in some situations, so untreatable epilepsy is one example, surgical severing or cutting the corpus callosum is actually undertaken as a last resort. So in the case of epilepsy, this is done to stop seizures spreading across the brain. So if a seizure begins in one particular region of the brain where you have excess electrical activity, the thought is that if you sever this corpus callosum, it won't just spread to other regions of the brain. So lastly, we're going to look at the different lobes of the brain. So I will ask you to be able to identify each of the lobes of the brain and provide a function. Now keep in mind that each lobe is going to have many, many different functions. I do not expect you to remember them all. I would simply pick one that stands out for you and keep that one in mind. So each cerebral hemisphere can be further divided into several lobes, and the lobes are named after the bones of the skull that protect them. So for example, our frontal lobe is here. It sits underneath our frontal bone. We have our occipital lobe at the back, and that's where our occipital bone of the skull would sit. So the frontal lobe is the most anterior part of the brain. It's this section highlighted here in blue. It's separated from the parietal lobe by the central sulcus, so a little groove here, and from the temporal lobe, which is this one here, by a lateral fissure, so a much deeper groove. In the pre-central gyrus, so the bump or fold just prior to the central sulcus, sits our primary motor cortex. And we will talk about this region of the brain in more detail in a future learning objective, but this is the region of the brain that initiates all voluntary motor movement, so all of our skeletal muscle movement. The frontal lobe also contains areas related to speech, personality, emotion, and cognition. Compared to other animal species, the frontal lobe of humans is considered very well developed, both in terms of its overall size and the kind of folding and wrinkling and therefore the number of neurons in the frontal lobe. And it's this that is believed to be what sets us apart from other similar animals in terms of our intellect. Moving posteriorly, here is our parietal lobe. As I said, it's separated from our frontal lobe by our central sulcus and from our occipital lobe by the parieto-occipital sulcus. In the post-central gyrus, so the fold or the ridge just posterior to the central sulcus, is our primary somatosensory cortex. Again, we will look at this in more detail in future, but this region receives all of the somatic sensory information of the whole body. The parietal lobe also contains several association areas that give meaning to sensory information. So for example, the primary somatosensory cortex will receive information that you can feel your phone in your pocket, but an association area will allow you to recognize that shape as a phone without actually needing to look at it. The occipital lobe, so this lobe here in pink, sits at the very back or the posterior of the brain, separated from the parietal lobe by that parieto-occipital sulcus, and then separated from the cerebellum here by our transverse fissure. The occipital lobe houses our primary visual cortex, so it receives all the information from our eyes here directly back into our occipital lobe. The occipital lobe also contains several association areas, so these are specialised for different visual tasks, such as visual spatial processing, colour differentiation, and motion perception, so how things move. Lastly, we have our temporal lobes, so that's this section here in green. We have one temporal lobe on each side of the brain, just above the ears. The temporal lobes are separated from our frontal lobe by that lateral fissure, so it's a bit of a deeper groove. The temporal lobes house structures that are important for long-term memory, one of which is the hippocampus. The temporal lobes also process auditory information in the primary auditory cortex as well as association areas. So this allows us to hear sounds, but then also recognise or understand what those sounds mean. Now, there are three other structures that sit within the cerebrum that I want to point out to you. These are not structures that you'll be assessed on, but they are structures that, depending on what degree you're doing, because remember there's lots of different degrees in regional anatomy and physiology, they might become important in future units. So I think it's really important to at least mention them. So firstly, deep within each cerebral hemisphere, so both on the left and the right side of the brain, we have three nuclei that are collectively termed the basal nuclei. So remember, nuclei is a collection or a cluster of cell bodies within the central nervous system. These three nuclei are the caudate nucleus, the putamen, and the globus pallidus. So our basal nuclei are involved in movement. They help initiate and terminate movement. They help regulate muscle tone. And they help control subconscious contractions of skeletal muscle, such as when you swing your arms when you're walking, which you don't really have to think about, or when you kind of hunch over laughing in response to a joke. So basal nuclei, really important for our movement. We then have the limbic system, and so all of these structures highlighted in green here are part of our limbic system. The limbic system is often referred to as our emotional brain because it plays a role in a range of different emotions, so things like pain, pleasure, affection, and anger. It's also involved in olfaction and memory, or smell and memory. So think about when you have a particular memory come up, when you smell something that your grandma used to cook, or you smell the perfume of someone you love. This area is also often studied when something is malfunctioning. So for example, a person whose amygdala is damaged often fails to recognize fearful expressions in others or express fear in situations where typically they should. This area was at one point in time quite often studied in criminals like serial killers. Along with other parts of the cerebrum, this area also plays a role in memory, and damage to this region can cause memory impairment. So speaking of memory, I also wanted to point out the hippocampus. So the hippocampus sits one in each temporal lobe, and from side on, from a lateral view, it does resemble the shape of a seahorse. Now, the hippocampus is important for two different types of memory. So declarative memory, which is your long-term memory, and also spatial memory. So declarative memory helps us learn something. It takes your information from short-term memory that you've just memorized to your long-term memory where you actually know it. So it's when you actually learn the bones or the names of the bones in the lab, or when you learn someone's name that you can just easily recall. Spatial memory involves pathways and navigation, so such as learning to drive to and from work, the path that you should take, or how to walk to and from the coffee shop. Now, neurons in the hippocampus actually continue to develop throughout adulthood, which is one of the very few regions of the brain where this happens. Unfortunately, however, this area is also typically the first and the most affected region in diseases like Alzheimer's. And you can see that in this image here. So in the image on the left, so image A, this is a patient with severe Alzheimer's. And you can see down here, which is where our hippocampus will usually sit, that area has been atrophied, or it is withered away. If you look on the right-hand side of the screen, this is a healthy person. Where the hippocampus is, that brain tissue is still intact. So moving to the next gross brain region, we have the diencephalon. This is the region highlighted in green. The diencephalon forms this core component of brain tissue, and it extends from the cerebrum down to the brainstem. The diencephalon contains three smaller structures: the thalamus, the hypothalamus, and then the epithalamus. So the thalamus is a large mass of grey matter located in the centre of the brain. Axons project out of the thalamus to the cerebral cortex in all different directions. Now, the primary function of the thalamus is to act as a relay station. So we have sensory impulses that reach the thalamus that then get sent out to the primary sensory cortex. It's also a relay station for our motor information. So it takes motor information from the cerebellum and the basal nuclei, also the primary motor cortex, and then sends it out to different areas of the cerebrum. Now, the thalamus is made up of several groups of nuclei on each side of the thalamus. So that's what each of these different colours is trying to represent. And they're grouped in clusters of neurons with similar positions on the thalamus and similar functions. So each group of nuclei will receive input and send output to similar areas or regions of the brain and transmit nerve impulses that have a similar function. So for example, the medial geniculate nuclei relays all of the auditory impulses for hearing from the ears to the primary auditory area of the cerebral cortex. The lateral ventral nuclei receives all of the input from the cerebellum and the basal nuclei and sends the output to the motor areas of the cerebral cortex. Next is the hypothalamus. So if here is our thalamus, this section just inferior and a bit anterior is our hypothalamus. The hypothalamus controls many different body activities, and it's known as one of the major regulators of homeostasis. Many different sensory impulses actually arrive at the hypothalamus, such as those for vision, taste, smell, stretch, touch, and pressure, as well as those that monitor the internal environment, so information regarding osmotic pressure, blood glucose levels, hormone concentration, and the temperature of the blood. It is the job of the hypothalamus to regulate these variables to ensure that the body remains in a state of homeostasis. The hypothalamus is also connected to the pituitary gland, so that's this little shape here. The pituitary gland is part of our endocrine system, and the hypothalamus produces hormones that either act on the pituitary gland or are produced in the hypothalamus and then released in the posterior pituitary gland. Lastly, we have the epithalamus. So the epithalamus is posterior to the thalamus, and it consists of two different structures, the pineal gland and the habenular nuclei. So the pineal gland is also part of the endocrine system. It secretes the hormone melatonin. So melatonin is produced and released in greater concentrations at night and is thought to promote sleepiness. When taken orally, melatonin can help reset the body's biological clock, and therefore it is used to help treat jet lag. The functions of the habenular nuclei are less understood, but they're thought to include the regulation of neurotransmitters serotonin and dopamine, which you may have heard of. They're involved in mood and motivation, help regulate the sleep-wake cycle alongside the pineal gland, and help integrate sensory information relating to olfaction or to smell. The next region is the brainstem. So the brainstem sits between the spinal cord and the diencephalon, and it consists of three structures: the midbrain, which is this section here, the pons, which is the rounded section, and then the medulla oblongata. So it's this second section here before we move into the spinal cord. So starting from the lower end of the brainstem, the medulla oblongata, which you can simply refer to as the medulla, is continuous with the spinal cord, and it forms the inferior part of the brainstem. It begins at the foramen magnum, so remember that hole in the base of the skull, and it extends to the inferior border of the pons. So when you see that rounded part of the brainstem. The white matter, which remember are our myelinated axons that are found in the medulla, contain all of the sensory and all of the motor tracts that extend between the spinal cord and other parts of the brain. The medulla also contains several nuclei, some of which control vital bodily functions. So some examples of these nuclei are the cardiovascular center and the medullary respiratory center. So the cardiovascular center regulates the rate and the force of the heartbeat, the diameter of the blood vessels. The medullary respiratory center adjusts the basic rhythm of breathing. Nuclei in the medulla also control reflexes for vomiting, swallowing, sneezing, coughing, and hiccuping. So we don't want to do damage to the medulla oblongata because it contains a lot of vital bodily functions. The pons then is moving superiorly, and it's the rounded kind of oval section that sits above the medulla. Pons means bridge, and as the name implies, the pons acts as a bridge connecting different parts of the brain with one another. Within the pons is the pontine respiratory group, which together with the medullary respiratory center helps control breathing. The pons also contains nuclei associated with 4 pairs of cranial nerves. So we have cranial nerves that exit both the medulla and the pons. Lastly, we have the midbrain, which is also referred to the mesencephalon as it's developing. It's the very top section of the brainstem, extending from the pons to the diencephalon. The cerebral aqueduct passes through the midbrain, which connects the third and the fourth ventricle. Now, on the anterior surface of the midbrain contain paired bundles of axons known as the peduncles. So the peduncles consist of axons of the corticospinal, corticobulbar, and corticopontine tracts. So we'll learn about these in the next lecture when we talk about the spinal cord, but they conduct nerve impulses from the motor areas of the brain to the spinal cord, the medulla, and the pons, respectively. The posterior portion of the brain is called the tectum, and it contains 4 rounded elevations. The 2 superior rounded elevations are nuclei known as the superior colliculi. The 2 inferior rounded elevations are known as our inferior colliculi. So the 2 superior colliculi are involved in reflexes to do with our vision. So things like tracking a moving object if you're looking at a car as it drives past you, or you're scanning a stationary image. So for example, you might be reading words on a page. Our inferior colliculi are to do with reflexes relating to sound or auditory activities. So for example, when you hear a really loud noise go off that startles you and you kind of jerk your head backwards, these reflexes occur in the inferior colliculi. And our last gross brain structure is the cerebellum. So the cerebellum is this structure here. Although the cerebellum accounts for only 10% of the brain's total volume, it contains 50% of the total number of neurons in the brain. So it kind of looks like the cerebrum, but much more highly folded and intricate. Now, the cerebellum receives information from sensory systems, the spinal cord, and other parts of the brain, and then it uses that information to regulate motor movements. So the cerebellum coordinates voluntary movements such as your posture, balance, coordination, and speech. And it's what allows us to have a smooth, coordinated, balanced muscular activity. So I think the best way to describe the function of the cerebellum is to actually show you a video of what happens when the cerebellum is functioning incorrectly.
Of tremor disorder, in addition to rest tremor and postural tremor, is intention action tremor seen with cerebellar disease. In this patient, who has had a traumatic hemorrhage involving one side of his cerebellar white matter pathways, he has a normal function on the right side, but the left side shows significant dysenergia and intention tremor. This type of tremor contrasts, however, with the postural tremor in that both the trajectory and the endpoint are abnormal. And most of this patient's dysfunction is in fact right at the end point when he tries to touch his nose or to bring his finger totally out. The trajectory is not normal, but the trajectory is not the area of maximal dysfunction. This type of tremor.
So what you can see there is with the patient's, right hand, it's a nice smooth movement. He can point in front of him and he can touch his nose. With his left hand, which is a side it's actually the opposite side of the brain, so the right side of his cerebellum is affected. He's a very shaky pointing and he can't quite get to his nose. So the cerebellum actually inhibits all of that extra movement that you see there in that shaky arm to create a nice, smooth, coordinated, balanced movement. Okay, so learning objective number 4 is now to locate and describe the sensory and the motor cortices. So this is an image looking down on the cerebrum. Here is our central sulcus, so that shallow groove separating the frontal lobe from the parietal lobe. Immediately anterior to that central sulcus is what we call the precentral gyrus. So it's precentral because it's just prior to the central sulcus, and it's agarist because it's a fold or a ridge. So it's this section highlighted in blue. This section is known as the primary motor cortex, also called the primary motor area or the M1. Now, in the primary motor area or cortex is a map of the entire body. So you can see that represented here. This is showing one hemisphere, so relating to one side of the body or movement to one side of the body. So what happens is that each region of the hemisphere or the cerebral cortex here relates to the voluntary contraction of a particular muscle or a particular group of muscles. So if we were to electrically stimulate at any particular point along this primary motor cortex, we would get a contraction in a muscle in the other side of the body. So you can see that the muscles are actually represented quite unequally in this cerebral cortex. So for example, our face has a lot of cerebral space, but our foot, our leg, our knee, our trunk has a much lesser space. This is because greater cortical area is devoted to those muscles involved in skilled, complex, or precise movement. So for example, we have a much larger cortical region devoted to muscles that move the fingers than the muscles that move the toes or the muscles that move the hip, even though the hip's a really big muscle region. So anything that requires really delicate precision, so writing your name, for example, is going to have a lot of dedicated cortical space. This distorted map of an individual is what's called the motor homunculus. So homunculus means little man. Immediately posterior to the central sulcus is our post-central gyrus. So post-central because it's posterior to the central sulcus, gyrus because it's another bump or ridge. And this is our primary somatosensory area or somatosensory cortex. And here we receive the nerve impulses from all of the sensory receptors that detect somatic sensations. So things like touch, pressure, vibration, itch, tickle, temperature, pain, proprioception. Like the primary cortex, we have a map of the entire body represented in that cortical area, with each region receiving impulses from a particular part of the body. The size of the cortical area receiving the impulses from a particular part of the body depends on the number of receptors in that area rather than the size of the body part. So for example, we have a large amount of cortical space dedicated to our lips compared to, say, our neck, our arm, or our elbow. Our lips are very sensitive. This is what allows us to talk nice and smoothly. This distorted image of our sensory body is called the sensory homunculus. In terms of our other senses, so our special senses, our sight, our hearing, our taste, our smell, we don't receive this information here in the primary somatosensory cortex because we have a primary visual area, we have a primary auditory area, a primary gustatory area, and a primary olfactory area. So therefore, these neurons do not transmit information to the primary somatosensory cortex. All right, so coming towards the end, I know this is a long one, I'm sorry. Our second last learning objective is to define the term association area and provide an example of an association area. So an association area is an area of the cerebral cortex which works to link and coordinate sensory and motor areas. So an association area allows us to produce a meaningful perception of the world. The functions of each association area are very specific. So what I've done is just highlighted a few different ones for you. So first off, our somatosensory association area is an area just posterior to the primary somatosensory cortex. It receives input from that sensory cortex as well as the thalamus and other regions. And this area allows you to determine the exact shape and texture of an object just by feeling it. So like I said previously, you can tell that your phone is in your pocket without needing to look at it. We then have Wernicke's area. So Wernicke's area is quite a big region of the left temporal and parietal lobe. And this area interprets the meaning of speech by recognizing the sounds of spoken words. So it allows you to understand what is being said. It's also active as you put your thoughts in your head into words, so as you go to try and say something. So those who have damage to this area can actually still speak, but they cannot arrange a sentence in a coherent fashion. Our visual association area is located in the occipital lobe. It receives sensory information from the primary visual area and also the thalamus. And it relates past and present visual experiences so that we can recognize an object simply by looking at it. It's not new information every time we see something. The auditory association area is located inferior and posterior to the primary auditory area in the temporal lobe. This allows you to recognize a particular sound or noise as, say, speech or music or a bird chirping. The prefrontal cortex, which is sometimes also called the frontal association area, is a really big component of the frontal lobe. It's particularly well developed in humans. And the prefrontal cortex is concerned with a person's personality, their intellect, their complex learning abilities, their initiative, reasoning, mood, and their conscience. We have Broca's speech area, which is also known as the motor speech area. So this is an association area found in the frontal lobe. Broca's area coordinates breathing and muscle contraction to allow speech. So unlike damage to Wernicke's area, individuals who do damage to Broca's area are unable to speak. So they can construct logical sentences in their mind, but they can't physically get those words out. Lastly, we have a premotor area. So it's a motor association area immediately anterior to the primary motor cortex. Neurons in this area communicate with the primary motor cortex, the sensory association areas, the basal nuclei, and the thalamus. And it helps you deal with learned motor activities of a complex and sequential nature. So for example, the specific groups of muscles that contract in a particular sequence as you sign your name, or you complete a skilled sporting move that you've practiced over and over. Now our last learning objective is to simply list the 12 cranial nerves. So cranial nerves are 12 pairs of cranial nerves that exit the brain or the brainstem. Each cranial nerve has both a number designated by a Roman numeral, so you will need to know Roman numerals up to 12, as well as a name. So the number indicates the order from anterior to posterior in which the nerves arise from the brain. The name designates either the distribution of that nerve or the function of that nerve. 3 of our cranial nerves are considered special sensory nerves as they carry axons of sensory neurons associated with our special senses. 5 of our cranial nerves are motor nerves because they contain only motor neurons. And then the 4 remaining cranial nerves are what we call mixed cranial nerves because they contain both axons of sensory neurons and axons of motor neurons. So we're not going to go through each cranial nerve individually because for most of you this is not something that will be of use to you later on in your degree, but we're just going to look at a few examples as well as go through a way to easily remember each of the cranial nerve names. So starting with cranial nerve number 1, which is our olfactory nerve. As the name suggests, this is related to our olfaction or our smell, and it's an entirely sensory nerve containing only axons that conduct nerve impulses from our nasal cavity back to our brain for smell. The olfactory nerve sits in the very anterior of the brain. So you can see that here. It extends forward and then it actually projects its little receptors into the nasal cavity through the olfactory foramina, which are the little holes in the cribriform plate on the ethmoid bone. Cranial nerve number 2 is our optic nerve. As you can probably guess, this is also an entirely sensory nerve. It conducts the nerve impulses for our vision. An optic nerve runs from the back of the eyeball to the middle of the brain where it forms a bit of a cross. That cross is called the optic chiasm. Half of the axons then cross over to the other side. The optic tract then runs back towards the occipital lobe. The next cranial nerve that we'll look at is cranial nerve number 8. It's called our vestibulocochlear nerve. So this is also another sensory nerve, but this one has 2 different branches, and you can kind of gather that from the name. It has a vestibular branch and a cochlear branch. So the vestibular branch carries nerve impulses for equilibrium, which come from receptors in the inner ear, and we'll talk about those towards the end of the semester. And then the cochlear branch carry impulses from our cochlea, which are the nerve impulses from hearing. We then have cranial nerve number 11, which is our accessory nerve. This is a purely motor nerve. The accessory nerve is also branched, and it transmits nerve impulses from the brain to our sternocleidomastoid muscle and our trapezius muscle to coordinate our head and neck movements. The last cranial nerve that we'll look at is cranial nerve number 7, which is a facial nerve, and this is one of our mixed cranial nerves. So its sensory axons extend from the taste buds in the tongue and then also the skin in the ear canal, and they relay sensory information relating to touch, pain, and thermal sensations back to the brain. Our motor axons innervate the middle ear, the face, the scalp, the neck muscles. And the facial nerve is particularly impressive because it innervates more named muscles than any other nerve in the entire body. Damage to the facial nerve is actually what causes Bell's palsy, which is a condition where you get paralysis in some of the face. So you can get a loss of taste, decreased salivation, and a loss of the ability to close your eye even when you're asleep. So lastly, as I mentioned at the start of this learning objective, we're not going to look at each cranial nerve in detail because for most of you, this is not something that will be important for your degree. For those who end up needing to know this information, you will cover this information in future units. What is important for this unit is understanding that there are 12 pairs of cranial nerves, that they are named based on their position and their function, and whether they're a sensory nerve, a motor nerve, or a mixed nerve. Now, one way to remember this information, which may or may not work for you, is to use a mnemonic. So a mnemonic are songs, rhymes, acronyms, or a sentence where you use the first letter of each of the words you want to remember. So for example, to remember the names of each of the cranial nerves in order, you might want to remember the sentence, on, they travelled and found Voldemort guarding very ancient Horcruxes. So the first letter of each of those words relates to the first letter of each of the cranial nerves. Obviously, this doesn't give the entire name away, but it does give you a hint as to what the name is and the order of those nerves. Also, this is a safe Harry Potter themed mnemonic that I decided to use. If you Google your own mnemonics and cranial nerves, there's millions that will come up on Google. Most of them are quite dirty and I couldn't use in the lecture, but find one that works for you. I tend to find that the stupider or the ruder the memory tool, the better I am able to remember it. Similarly, with whether the nerve is sensory, motor, or both, I find mnemonics really useful. So Disa, for example, told me this mnemonic when I first moved to Canberra about 10 years ago. So some say marry money, but my brother says big boobs matter more. So S is for a sensory nerve, M is for a motor nerve, and B is for both. So our olfactory nerve, for an example, is a sensory nerve. Okay, so again, I apologise for how long this lecture is. There's lots to cover in the brain. Please note that I am aware that this lecture had lots of content to cover. There's lots of different regions, lots of different functions, some that are really obvious and some that we are more vague about. When we come to assess this content, that will be taken into consideration. So I'm not going to ask you to, say, identify the habenula nuclei and provide a function or describe, the input and the output of the prefrontal cortex. A lot of this information is going to be important for you to be aware of as you move through your degree, not necessarily something that you're going to be tested on. So just as a reminder, I've provided a summary of many of these structures that we've covered today, where they're located and what they do on Canvas. But if you have any other questions, please let me know. Thanks again for listening, and I will see you in the lab.